Glucose metabolism refers to the processes through which the body manages glucose, a simple sugar, for energy. It involves several key steps and pathways, including glycolysis, the Krebs cycle, also known as the citric acid cycle, and oxidative phosphorylation. Here's an overview of these stages. 1. Glycolysis. This occurs in the cytoplasm of cells and is the process where one molecule of glucose, 6-carbon sugar, is broken down into two molecules of pyruvate, 3-carbon compounds. Glycolysis generates a small amount of ATP energy and NADH, an electron carrier. 2. Pyruvate oxidation. Pyruvate molecules produced in glycolysis enter the mitochondria, where they are converted into acetyl-CoA, releasing carbon dioxide CO2 and producing NADH. Acetyl-CoA is the substrate for the next step. 3. Krebs cycle, citric acid cycle. Acetyl-CoA enters the Krebs cycle within the mitochondria, where it is broken down into CO2. This cycle produces ATP, NADH, and FADH2, another electron carrier. 4. Oxidative phosphorylation, electron transport chain. NADH and FADH2 produced in earlier steps donate electrons to the electron transport chain in the inner mitochondrial membrane. This process drives the production of a large amount of ATP as protons are pumped across the membrane, creating a gradient that powers ATP synthesis. Oxygen acts as the final electron acceptor, forming water. 5. Gluconeogenesis. In conditions where glucose is scarce, e.g., fasting, the body can produce glucose from non-carbohydrate sources like amino acids, lactate, and glycerol through gluconeogenesis, mainly occurring in the liver. 6. Glycogen metabolism. The body stores excess glucose as glycogen, primarily in the liver and muscle cells. When glucose levels fall, glycogen can be broken down through glycogenolysis to release glucose into the bloodstream for energy. Glucose metabolism is tightly regulated by hormones like insulin and glucagon. Insulin facilitates glucose uptake by cells, promoting storage as glycogen and reducing blood glucose levels. Glucagon, on the other hand, stimulates glycogen breakdown and glucose production, increasing blood glucose levels. Disruptions in glucose metabolism, such as in diabetes mellitus, can lead to impaired energy use and hyperglycemia, high blood glucose. The complete metabolism of one molecule of glucose through cellular respiration yields a significant amount of energy, primarily in the form of ATP, adenosine triphosphate. Here's a breakdown of how much ATP is produced in each stage of glucose metabolism. 1. Glycolysis. ATP produced, 4 ATP. ATP consumed, 2 ATP. Net ATP gained, 2 ATP. NADH produced, 2 NADH. Each NADH can yield about 2.5 ATP during oxidative phosphorylation, resulting in a total of 5 ATP. 2. Pyruvate oxidation, 2 pyruvate molecules per glucose. NADH produced, 2 NADH, 1 per pyruvate, yielding 5 ATP. 3. Krebs cycle, citric acid cycle, 2 acetyl-CoA per glucose. ATP produced, 2 ATP, 1 per acetyl-CoA. NADH produced, 6 NADH, yielding 15 ATP. FADH2 produced, 2 FADH2, yielding 3 ATP. 4. Oxidative phosphorylation, NADH yields. Each NADH yields approximately 2.5 ATP. Each FADH2 yields approximately 1.5 ATP. Total ATP yield from one glucose molecule. Combining the energy yields from each stage, the approximate total ATP production is glycolysis, 2 ATP net plus 5 ATP from 2 NADH equals 7 ATP. Pyruvate oxidation, 5 ATP from 2 NADH. Krebs cycle, 2 ATP plus 15 ATP from 6 NADH plus 3 ATP from 2 FADH2 equals 20 ATP. Total ATP yield, 7 plus 5 plus 20 equals 32 ATP. In reality, the ATP yield can vary slightly due to inefficiencies in the electron transport chain and transport of NADH into the mitochondria. Therefore, the total is often estimated to be between 30 to 32 ATP per molecule of glucose.